Antigens and antibodies interact with each other to form immune complexes. The antibodies bind to the antigens in order to inactivate them. The immune system fights against pathogens by this mechanism and prevents us from diseases. Antigens are usually the markers of pathogens which are identified by the cells of immune system. The antibodies bind to it and initiate the effector mechanisms which lead to the destruction of antigen carrying invading pathogens. The antigens and antibodies are held together by non-covalent bonds in immune complex. They bind to each other through van der Waals forces, hydrophobic interactions, hydrogen bonds and electrostatic bonds. These forces of attraction are comparatively weaker than the covalent bonds, but their cumulative effect is enormous, which leads to the destruction of target molecules. The binding between the antigens and antibodies is highly specific. This specificity is the selective attachment of the molecules to complementary structures. The site of attachment on antigen is epitope and antibody is paratope. Their molecular structures contain placement of complementary shapes and bonds. Formation of the immune complex. Antigen antibodies interact to form a lattice which grows in size and forms an immune complex. The immune complex will be formed only if the antibodies are bivalent and the antigens are multivalent. This leads to the sharing of antigen antibody valencies, leading to their cross linking and increase in the size of lattice. If the antibodies are monovalent, there will be no sharing of valencies, therefore, no lattice formation. The antigens should have some multiple epitopes of same type to bind with more than one antibody and increase the size of lattice. The association between antigen and antibodies is governed by the strength of the bonds between them. If the association is powerful, the formation of immune complex will proceed. This strength is measured in terms of affinity and avidity. Affinity. Affinity is the measure of the strength of the individual bonds between antigens and antibodies. This helps in their attachment. This binding strength leads to the sustenance of antigen antibody complex and increase in the size of lattice. Formation of large size lattice leads to the observable immune complex. Avidity. Avidity is the overall strength of the immune complex, which involves the force of attraction between all the antibodies and the antigen molecules in the complex. This includes different types of antibodies bound to the multiple types of epitopes present in the immune complex. Avidity is an important aspect because if the affinity between the individual bonds is less, the higher avidity will lead to the powerful binding in immune complex. The antigens and antibodies form observable immune complex only if their valencies are shared and cross-linking occurs. This in turn depends on the number of antigens and antibodies present in the reaction mixture. This can be further understood by an experiment based on zone phenomena. If we take a series of test tubes with equal quantity of antibodies in all of them and then add increasing concentrations of its complementary antigens in all the tubes such that the starting tubes contain lesser quantity of antigens and the later ones contain higher concentration. The immune complex will be formed in all the tubes because the antigens and antibodies are specific for each other, but the size of complex will vary greatly. In the earlier tubes where the antibodies are in excess, the antigens are available for sharing of valencies. This hinders the growth of size of lattice. Therefore, small complexes are formed. This is known as the zone of antibody excess or prozone. In the later tubes, where the antigens are in excess, there are numerous epitopes available for the binding to antibody paratopes. In this case, both the paratopes of an antibody molecule are satisfied by different antigens. So, there is no cross linking again. Due to small size of lattice, observable immune complex is not formed. This is known as zone of antigen excess or post zone. In case of the middle tubes, large lattice is formed. The valencies of antigens and antibodies are nearly equal in these tubes. The size of lattice increases due to extensive sharing of antigen antibody valencies, which leads to large immune complexes. No small immune complexes are formed in these tubes. This is known as zone of equivalence. There are two important attributes of any antigen antibody reaction: specificity and sensitivity. Specificity of the antigen antibody reaction refers to the specific binding of an antigen to only its complementary antibodies and not with the one which does not have complementary structure on its paratope. The presence of other antibodies does not interfere in the complementary binding. Sensitivity. Sensitivity of the reaction means its ability to form immune complex even at low concentrations of antigens and antibodies. If antigen antibody concentration is less, the observable immune complex formation is difficult. The immune complex can be observed when the valencies of antigens and antibodies are equal. Observable immune complex forms only if there is sharing of valencies. Cross reactions. In majority of the cases, antibodies bind to their specific antigens, as it is well known that the binding occurs due to the presence of complementary structures. However, sometimes the antibodies may bind to another molecule possessing similar structure. Two different types of molecules may have similar epitopes. The similarity in the epitopes may be to varying extents. Strength of the binding depends upon the degree of similarity. It is observed that some pathogens possess antigenic epitopes similar to normal host cell components. Thus, infection of these pathogens causes cross reactivity, which may further lead to autoimmune disorders. Some commonly found bacteria in intestine carries antigens similar to the blood group antigens. Streptococcus pyogenes possesses cell wall protein M antigens which cross react with several myocardial and skeletal muscle proteins. This may lead to heart and kidney damage following streptococcal infections. Vaccination process is a useful application of cross reactivity. Jenner vaccinated people against smallpox using cowpox vaccinia virus. Types of antigen antibody reactions. Antigen antibody reactions are of two types: agglutination and precipitation. The type of reaction taking place depends on the nature of antigen. Agglutination: when a particulate antigen reacts with its complementary antibody in the presence of suitable electrolytes at optimum pH and temperature, an observable immune complex is formed. The immune complex appears in the form of clumps. This is agglutination reaction. Agglutination occurs usually when the antigens are large molecules in the suspension, for example, bacteria or red blood cells. The antibodies that produce this kind of reaction are known as agglutinins. Precipitation: when a soluble antigen reacts with its complementary antibody in the presence of suitable electrolytes at optimum pH and temperature, an observable immune complex is formed. This is precipitation reaction. The antibodies that aggregate soluble antigens are known as precipitants. The antigens involved in precipitation reaction are usually of smaller size. Sometimes the precipitate formed due to soluble antigen antibody complex keeps floating in the liquid. This is known as flocculation reaction. Serological reactions. Antigen antibody reactions taking place in vitro are known as serological reactions. Diagnosis of diseases by serological reactions is one of the preliminary tests for patients suffering from infections. Agglutination and precipitation are the basic types of antigen antibody reactions, which have immense medical applications in the form of serological reactions. Immune complex formation forms the basis of serodiagnostic tests. Serodiagnostic tests are used for the diagnosis of infectious diseases. Blood sample is withdrawn from the patient and serum is recovered. The serum contains the antigens of the microorganisms with which the patient is infected. Presence of these antigens can be detected in the patient serum. The patient serum also contains antibodies against these antigens. The antibodies are produced in the patient's body in response to the antigens on the pathogenic agent. Diagnosis of the disease can be done by detecting the presence of antigen or antibody in the patient serum by agglutination or precipitation reactions. A diagnostic test has to be efficient enough to detect the presence of even low levels of pathogenic antigens present in the patient serum, that is sensitive, and also accurate strain of the pathogen, that is specific. Agglutination reactions are more sensitive than precipitation reactions. The difference in agglutination and precipitation is only because of the nature of the antigen. If the antigen is particulate, the reaction is agglutination, and if the antigen is soluble, the reaction is precipitation. Precipitation reactions can be converted to agglutination reactions to increase the sensitivity. This conversion is done by increasing the size of the antigen molecule. The antigen molecules can be bound to inert substances like polystyrene. Numerous antigens can be coated on a single latex bead. Sensitivity and specificity are inversely proportional to each other. If a reaction is specific, which means the antibodies will bind to the antigens only if complementarity is perfect and not otherwise. Now, if the complementary antibodies are not in more number, observable immune complex will not be formed, which means that even in the presence of antibodies and antigens, there is no formation of immune complex. This is false negative test. We have made the reaction essentially specific, which has reduced the sensitivity of the reaction. If we want to increase the sensitivity of the test, specificity will be compromised. If we want to detect very low levels of the antigen or a pathogenic agent bearing that antigen, we will have to use the antibodies against all the epitopes of the antigen or all the antigens of the pathogenic agent in consideration. Using so many different types of antibodies makes the process non-specific. This problem prevails in the application of antigen-antibody reactions used for the diagnosis of infectious diseases. To overcome this problem and increase the efficiency of the test, some new techniques have been developed which exhibit high sensitivity as well as specificity. Serological reactions can be qualitative or quantitative. Qualitative tests are done to check the presence or absence of the infectious agent, which in turn confirms the presence or absence of the disease. Example, VDRL test for syphilis and Vidal test for typhoid. Quantitative tests are done to find out the severity of the disease. Higher the number of antibodies in the serum, severe is the disease. That is, high number of infectious agents are present in the patient. The quantification is done by determining its titer value. Both precipitation and agglutination reactions can be used for diagnosis by following methods. Precipitation reactions: slide test, tube test and immunodiffusion. Immunodiffusion is further categorized into single dimension single diffusion, single dimension double diffusion, double dimension single diffusion and double dimension double diffusion. Next is agglutination reactions: slide agglutination, tube agglutination, hemagglutination and passive agglutination test. Passive agglutination test can be further divided into passive hemagglutination and latex agglutination test. Precipitation reactions: slide test. A drop of antigen suspension and antiserum is placed on a glass slide. They are mixed to facilitate the immune complex formation. If the antiserum contains antibodies against the antigen used, immune complex will be formed. Example: VDRL test for the diagnosis of syphilis. This is the qualitative test for the diagnosis. Tube test. Tube test is usually done for quantitative diagnosis of any disease. Flocculation test performed in tubes for the diagnosis of syphilis is known as Kahn test. Standardization of toxins and toxoids is also done by tube flocculation test. Immunodiffusion. Immunodiffusion reactions are the antigen antibody reactions in gel. The antigens and antibodies are allowed to react in gel, which results in the formation of precipitate lines. These tests can be qualitative or quantitative. Single dimension single diffusion. Antigens and antibodies are allowed to react in agarose gels in a test tube. Antibodies or antiserum is incorporated in the agarose gel and placed in a test tube. The antigen suspension is poured on the top of the test tube. It is then allowed to diffuse down through the gel. A line of precipitation is formed where the antigen and antibodies interact in sufficient number to give observable reaction. In this process, only antigen is moving, that too in single dimension, that is downwards. Therefore, it is known as single dimension single diffusion. Single dimension double diffusion. The antibodies are mixed with agarose in a test tube, above which is layered plain agar. Antigen suspension is placed above the plain agar. The antigens move downwards and antibodies move upwards. They meet in the plain agar and form a line of precipitation if they are complementary to each other. This is known as double diffusion because both antigens and antibodies are moving, but only in one dimension. Double dimension single diffusion. This is performed on a glass slide. The antibodies are incorporated in the agarose and plated as a thin film on a glass slide. After the agarose layer solidifies, wells are cut in the agar. Antigen suspension is added to these wells. The antigens diffuse out of these wells and start interacting with the antibodies as soon as they enter the agarose layer. The diffusion continues and the size of antigen antibody lattice increases till observable zone of precipitation is formed around the wells. Occurrence of the precipitation zone is indicative of positive test, and the size of the zone indicates the quantity of the antigen present in the test sample. Therefore, this is qualitative as well as quantitative. This is also known as radial immunodiffusion, as circular precipitin bands are observed. The method was developed by Badsini. Double dimension double diffusion. This technique was given by Octoloni and is also performed on slide. Plain agar is layered on a glass slide. Wells are cut in the agar. Antigens and antibodies are put in separate wells. They diffuse towards each other, which leads to the immune complex formation and the appearance of a precipitation line. This technique is applied for analyzing the type of antigen present in the given sample. The two antigens in consideration are placed in nearby wells and the antibody is placed in the opposite well. Immune complex may be formed between the wells if antigens and antibodies recognize each other. The line of precipitation may merge or cross each other or form a spur. The pattern depends upon the relative nature of the antigens. If the two antigens placed in nearby wells are completely identical, the lines of precipitation will merge and the pattern is known as pattern of identity. If the two antigens are dissimilar, the line of precipitation will cross each other. This is known as pattern of non-identity. In the third case, the antigens have some similar epitopes and some different. Therefore, spur formation takes place. This is known as pattern of partial identity. Agglutination reactions. Slide agglutination. A drop of particulate antigen is mixed with the serum containing antigens on a glass slide. If the antigens and antibodies are complementary to each other, clumping will occur. The formation of agglutinates depends on the concentration of antigens and antibodies. The valencies of both should be equal. In case of antigen or antibody excess, the agglutinates will not be observed properly. The test should be conducted in the presence of a positive and a negative control. The positive control will show the formation of appropriate agglutinates and the negative will rule out the possibility of autoagglutination of antigens. Blood grouping and Vidal test for the diagnosis of typhoid are examples of agglutination reaction. Tube agglutination. Agglutination reaction in tubes is usually done for quantitative estimation of antibodies in patient serum. The patient serum is added to a series of tubes in decreasing concentration. Two-fold serial dilution of serum is done. For example, the first tube will contain original concentration of the serum, second will contain 1:2 diluted serum, third will contain 1:4 diluted serum, then 1:8, 1:16, 1:32, 1:64, 1:128 and so on. The antigen added in all the tubes is in same quantity. The agglutination is observed in the tubes when the concentration of antigen and antibodies lie in the range of equivalence. The last tube, which shows agglutination, represents the titer value. Suppose agglutination is observed in the tube containing 1:32 diluted serum and not in 1:64 diluted serum, then the titer value is 1:32. This indicates the severity of the disease in the patient. Higher the titer value, severe is the infection. Titer value for a patient suffering from typhoid is determined by using flagellar H antigen or somatic O antigen of Salmonella typhi. Hemagglutination. Hemagglutination is a process in which agglutination of red blood cells occur. Blood grouping is an example of hemagglutination. Blood sample is treated separately with anti-A, anti-B and anti-Rh antibodies. The RBCs carrying antigen A will agglutinate with anti-A antibodies. Those carrying antigen B will agglutinate with anti-B antibodies, and Rh antibodies will agglutinate the RBCs carrying Rh antigens. Presence of Rh antigens is denoted by positive sign and its absence is denoted by a negative sign. Agglutination of RBCs is hemagglutination. Therefore, if any other antigen binds on the RBC surface and leads to its agglutination, we call it hemagglutination. Passive agglutination test. When agglutination process is facilitated by coating of soluble antigens on a large carrier molecule, it is known as passive agglutination test. The carrier can be RBCs or polystyrene beads. When antigens are coated on RBCs for test, the process is known as passive hemagglutination, and when polystyrene beads are used, it is known as latex agglutination test. Passive hemagglutination. Soluble antigens can be coated on RBCs, which increase the size of the antigens and converts precipitation reaction into more sensitive agglutination reactions. The serum suspected of containing antibodies against these antigens are mixed with it and allowed to agglutinate. If hemagglutination occurs, the test is positive and the patient's serum is containing antibodies against the antigens coated on RBCs. The agglutination is because of some other antigens coated on RBCs and not its own antigens. Therefore, the technique is passive hemagglutination. Latex agglutination test. The coating of antigens or antibodies can be done on an inert material like latex beads, also known as polystyrene beads, in place of RBCs. This process enables the easy detection of the presence of antigens or antibodies in the patient's serum, urine, cerebrospinal fluid or saliva. It is commonly used for detecting the presence of rheumatoid factor in patient serum. Rubella virus, Haemophilus influenzae, Streptococcus pneumoniae and group B Streptococcus infections can also be detected by latex agglutination test. The latex beads agglutinate because of the antigens or antibodies coated on it, therefore known as latex agglutination test. So, we understood that antigens and antibodies interact with each other only if they are complementary to each other, which forms the basis of most important aspect, the application in the field of diagnosis of diseases.